Hello, science enthusiasts and curious minds. Today, we're embarking on an electrifying journey into the microscopic world of neurotransmission. We're Equation by Pharmacist Moms, and in this video, we'll unravel the fascinating story of chemical substances that serve as the messengers of the mind. Welcome to the captivating realm of synaptic transmitters. Our brains are a symphony of chemical signals, orchestrating everything from our thoughts to our movements. In this exploration, we'll dive deep into the diverse and dynamic world of neurotransmitters, understanding the key players that bridge the gaps between neurons and make communication within our brains possible. Whether you're a budding neuroscientist, a student eager to grasp the essentials, or simply someone intrigued by the magic within, this video is your ticket to unlocking the secrets of synaptic communication. So, hit that subscribe button, join our community of brain enthusiasts, and let's unravel the chemical symphony of the mind together. In this video, we will learn 1. Small molecule, rapidly acting transmitters, including recycling of the small molecule types of vesicles, classes of small molecule, rapidly acting transmitters, characteristics of some important small molecule transmitters, and 2. Neuropeptide, slowly acting transmitters or growth factors. Let's start off. What are the different chemical substances that function as synaptic transmitters? More than 50 chemical substances have been proved or postulated to function as synaptic transmitters. They can be categorized into two groups. One group comprises small molecule, rapidly acting transmitters. The other is made up of a large number of neuropeptides, of much larger molecular size, that are usually much more slowly acting. We will discuss them one by one. The small molecule, rapidly acting transmitters are the ones that cause most acute responses of the nervous system, such as transmission of sensory signals to the brain and of motor signals back to the muscles. In most cases, the small molecule types of transmitters are synthesized in the cytosol of the presynaptic terminal and are absorbed by means of active transport into the many transmitter vesicles in the terminal. Then, each time an action potential reaches the presynaptic terminal, a few vesicles at a time release their transmitter into the synaptic cleft. This usually occurs within a millisecond or less. The subsequent action of the small molecule type of transmitter on the membrane receptors of the postsynaptic neuron usually also occurs within another millisecond or less. Most often, the effect is to increase or decrease conductance through ion channels. An example is to increase sodium conductance, which causes excitation, or to increase potassium or chloride conductance, which causes inhibition. How small molecule types of vesicles are recycled? Vesicles that store and release small molecule transmitters are continually recycled and used over and over again. After they fuse with the synaptic membrane and open to release their transmitter substance, the vesicle membrane, at first, simply becomes part of the synaptic membrane. However, within seconds to minutes, the vesicle portion of the membrane invaginates back to the inside of the presynaptic terminal and pinches off to form a new vesicle. And the new vesicular membrane still contains appropriate enzyme proteins or transport proteins required for synthesizing and or concentrating new transmitter substance inside the vesicle. Acetylcholine is a typical small molecule transmitter that obeys the principles of synthesis and release stated earlier. This transmitter substance is synthesized in the presynaptic terminal from acetyl coenzyme A and choline in the presence of the enzyme choline acetyltransferase. Then it is transported into its specific vesicles. When the vesicles later release the acetylcholine into the synaptic cleft, during synaptic neuronal signal transmission, the acetylcholine is rapidly split again to acetate and choline by the enzyme cholinesterase, which is present in the proteoglycan reticulum that fills the space of the synaptic cleft. And then again, inside the presynaptic terminal, the vesicles are recycled, choline is actively transported back into the terminal to be used again for synthesis of new acetylcholine. What are the classes of small molecule, rapidly acting transmitters? Let's learn. Class 1. It includes acetylcholine. Class 2, the amines. It encompasses norepinephrine, epinephrine, dopamine, serotonin, and histamine. Class 3, amino acids. It comprises of gamma-aminobutyric acid, that is, GABA, glycine, glutamate, and aspartate. Class 4. It incorporates nitric oxide. Now, we will discuss some of the most important of the small molecule transmitters in succession. First one is acetylcholine. Acetylcholine is secreted by neurons in many areas of the nervous system, but specifically by the terminals of the large pyramidal cells from the motor cortex. Several different types of neurons in the basal ganglia. The motor neurons that innervate the skeletal muscles. The preganglionic neurons of the autonomic nervous system. The postganglionic neurons of the parasympathetic nervous system, and some of the postganglionic neurons of the sympathetic nervous system. In most instances, acetylcholine has an excitatory effect. However, it is known to have inhibitory effects at some peripheral parasympathetic nerve endings, such as inhibition of the heart by the vagus nerves. Second one is norepinephrine. Norepinephrine is secreted by the terminals of many neurons whose cell bodies are located in the brainstem and hypothalamus. Specifically, norepinephrine-secreting neurons located in the locus coeruleus in the pons send nerve fibers to widespread areas of the brain to help control overall activity and mood of the mind, such as increasing the level of wakefulness. In most of these areas, norepinephrine probably activates excitatory receptors, but in a few areas, it activates inhibitory receptors instead. Norepinephrine is also secreted by most postganglionic neurons of the sympathetic nervous system, where it excites some organs, but inhibits others. Next one is dopamine. Dopamine is secreted by neurons that originate in the substantia nigra. The termination of these neurons is mainly in the striatal region of the basal ganglia. The effect of dopamine is usually inhibition. Upcoming is glycine. Glycine is secreted mainly at synapses in the spinal cord. It is believed to always act as an inhibitory transmitter. Next one is GABA, that is, gamma-aminobutyric acid. It is secreted by nerve terminals in the spinal cord, cerebellum, basal ganglia, and many areas of the cortex. It is believed always to cause inhibition. Following is glutamate. Glutamate is secreted by the presynaptic terminals in many of the sensory pathways, entering the central nervous system, as well as in many areas of the cerebral cortex. It probably always causes excitation. Adjacent is serotonin. Serotonin is secreted by nuclei that originate in the median raphe of the brainstem and project to many brain and spinal cord areas, especially to the dorsal horns of the spinal cord and to the hypothalamus. Serotonin acts as an inhibitor of pain pathways in the cord, and an inhibitor action in the higher regions of the nervous system is believed to help control the mood of the person, perhaps even to cause sleep. To come is nitric oxide. Nitric oxide is especially secreted by nerve terminals in areas of the brain responsible for long-term behavior and for memory. Nitric oxide is different from other small molecule transmitters in its mechanism of formation in the presynaptic terminal and in its actions on the postsynaptic neuron. It is not preformed and stored in vesicles in the presynaptic terminal, as are other transmitters. Instead, it is synthesized, almost instantly as needed, and it then diffuses out of the presynaptic terminals, over a period of seconds, rather than being released, in vesicular packets. Next, it diffuses into postsynaptic neurons nearby. In the postsynaptic neuron, it usually does not greatly alter the membrane potential, but instead changes intracellular metabolic functions that modify neuronal excitability for seconds, minutes, or perhaps even longer. Now, we'll learn the second group of transmitter substances that are neuropeptides. In contrast to small molecule transmitters, the neuropeptides usually cause more prolonged actions, such as long-term changes in numbers of neuronal receptors, long-term opening or closure of certain ion channels, and possibly even long-term changes in numbers of synapses or sizes of synapses. The neuropeptides are not synthesized in the cytosol of the presynaptic terminals. Instead, they are synthesized as integral parts of large protein molecules by ribosomes in the neuronal cell body. Because of this laborious method of forming the neuropeptides, much smaller quantities of them are usually released than of the small molecule transmitters. This is partly compensated for by the fact that the neuropeptides are generally 1,000 or more times as potent as the small molecule transmitters. Another important characteristic of the neuropeptides is that they often cause much more prolonged actions. Some of these actions include prolonged closure of calcium channels, prolonged changes in the metabolic machinery of cells, prolonged changes in activation or deactivation of specific genes in the cell nucleus, and/or prolonged alterations in numbers of excitatory or inhibitory receptors. Some of these effects last for days, but others, perhaps for months or years. Our knowledge of the functions of the neuropeptides is only beginning to develop. Neuropeptides include hypothalamic-releasing hormones. These include thyrotrophin-releasing hormone, luteinizing hormone-releasing hormone, and somatostatin, which is growth hormone inhibitory factor. Pituitary peptides. These incorporate adrenocorticotropic hormone, that is, ACTH, beta-endorphin, alpha-melanocyte stimulating hormone, prolactin, luteinizing hormone, thyrotrophin, growth hormone, vasopressin, and oxytocin. Peptides that act on gut and brain. These encompass leucine and kephalin, methionine and kephalin, substance P, gastrin, cholecystokinin, vasoactive intestinal polypeptide, that is, VIP, nerve growth factor, brain-derived neurotropic factor, neurotensin, insulin, and glucagon. Peptides from other tissues include angiotensin II, bradykinin, carnosine, sleep peptides, and calcitonin. And there you have it, our expedition into the mesmerizing world of synaptic transmitters. We hope this journey has illuminated the incredible diversity and significance of these chemical messengers in shaping our neural landscape. If you found this exploration of neurotransmitters intriguing, don't forget to give it a thumbs up, share it with your fellow science enthusiasts, and subscribe for more enlightening content on the inner workings of the brain. The world of synaptic transmitters is vast and ever-evolving, and there's always more to discover. Thank you for joining us on this neurochemical voyage. Your questions and engagement fuel our passion for unraveling the mysteries of the brain. Keep that curiosity alive, and remember, the more we understand about these chemical messengers, the more we understand about ourselves. We are Equation by Pharmacist Moms, signing off for now, but stay tuned for our upcoming videos as we continue to explore the intricacies of the brain's symphony. Until next time, keep those neurons firing and your curiosity thriving.